Okay, so Laura will be in charge of pausing the recording, and will tell us when to pause, Pooja, and we'll take questions during that period. Does that make sense?
Okay, sounds good. Yep.
Okay, please, I'll turn it over to you, Pooja. Go ahead.
Okay, thank you so much, Mark, for the introduction. So as Mark said, I'm going to be giving the Beyond the Classroom lecture on fermentation today. I'm sure some of you may know everything I'm going to cover today, so just bear with me, take it as a refresher. And I'm hoping at the end of the talk, everyone understands how we get the bacteria to do all the work for us. So it's pretty awesome. So I'll start with a safety moment. And I'm doing a safety moment on safe lifting. I think a lot of us order packages and we need to get it into the lab and also move a lot of stuff around in the lab. And I think it's really important to know how to safely lift heavy weights. So it's important to size up the load, get a good grip, keep it close to your body, and also probably the most important thing is to make sure you lift with your legs and you don't use your back, so you don't hurt your back, as I have done in the past. Moving on, this is kind of the outline of my presentation. I actually changed it a little bit, so it's now in 3 sections. But I think this should work just fine. So the first section is just an introduction of fermentation with a couple of historical examples of industrial fermentation. And then I'm going to quickly move on to the processes or the molecular biology techniques that are involved in converting the sugar to the chemical. So exactly what do we do in our lab that can convert the sugar to the chemical final compound. And then my second section is going to be on how I can illustrate these different processes using an example of a bioproduct synthesis in our lab. And I'm going to use 1,4-butane-diol to illustrate all of the different processes that I talk about. And finally, in my last section, I just have a few slides to show you what are the efforts being made in the industry to improve process yields and process titers, right from milligrams per liter to several tens of grams per liter. And I'll use penicillin as an example for this section. So my first section, again, is introduction and molecular biology techniques. So fermentation is actually derived from the Latin verb fervere, which means to boil. And this is because in ancient times, when if you leave crushed grapes for a long period of time in these large vessels, they would start bubbling, and it looked like it was boiling, and that's why the name came into being. And those bubbles were actually carbon dioxide that is released as a byproduct of fermentation. And the earliest chemical evidence of an alcoholic beverage was actually in China, in 7000 BC. So, I'm sorry. Yeah. So the earliest evidence of the alcoholic beverage was discovered in China in 7000 BC. So actually, fermentation, natural fermentation, even precedes human history. And it wasn't until 1800, so after several thousand years, that we actually started understanding the process of fermentation. So it was Pasteur in 1860 who suggested that it was the action of living microorganisms that actually cause fermentation. And he defined fermentation incorrectly, actually, as life without air. And it was incorrect at the time because today it includes both aerobic as well as anaerobic metabolism of microorganisms to produce energy from organic compounds. So this is a picture of some of the things that we eat every day, consume every day, that's a product of fermentation, ranging from bread, cheese, to different types of alcohol. And also recently, most of the prescription drugs, the antibiotics and the vaccines that we take are actually produced using fermentation in the pharmaceutical industry. So this slide shows you a simplified timeline of fermentation. This is really, really simplified, just to give you a picture of the time it has taken for us to reach where we are today. So starting in 7000 BC, we had the first evidence of alcoholic beverages like beer and wine, moving on to natural fermentation of bread. So During fermentation, the carbon dioxide that is released, that helps to leaven the bread. Moving further, cheese, vinegar, lactic acid, a lot of the compounds used in preserving food is actually produced as an act of fermentation. And in the 20th century, we had a huge expansion in the number of products produced using fermentation. So starting with organic acids in 1940s, including citric acid and gluconic acid, moving on to penicillin, which was the first antibiotic to be produced from fermentation. And then a lot of amino acids like lysine, glutamic acid, that's MSG, that we use in the food industry, is actually produced from fermentation. And more recently, we are moving into protein products using fermentation. So insulin was actually the first human protein to be produced using fermentation. And today, like I mentioned, a lot of the vaccines, a lot of the antibiotics are produced using fermentation processes. So I just want to give you 2 examples of fermentation ranging from something really simple.
Pooja, we lost your audio. Hang on 1 second, okay? Okay, try to say something now.
Can you hear me now?
Yes.
Okay. Okay, so I'm going to give you 2 examples of historically famous examples of fermentation. So the first one is going to be a really simple molecule, ethanol, which is the alcohol. And the second example is going to be insulin, which is a very complex protein. So just to give you a contrast of the different products that you can get using the same fermentation process. So I'm sure a lot of you are already aware of ethanol fermentation, but so in ancient times, if you leave fruits or grains in these specific jars, given the right temperature and humidity, it would then ferment over a period of time to produce alcohol. And over several thousand years, we now have a much better understanding of fermentation. So we now know that it's the action of a yeast or bacteria that causes fermentation. We also know the enzymes in these organisms that actually catalyze the reaction. So we are now in 2015, the worldwide production of bioethanol from corn and other sugars was about 25 billion gallons. So we've really come a long way. And this ethanol can be used in the paint industry. It's also used as a biofuel, E85. The picture below shows you a schematic of the pathway for ethanol production from glucose, which is one of the most common sugars present in different biomass feedstocks. So it's a 6-carbon sugar, and it first undergoes glycolysis, which is about 10 steps to give you pyruvate, which is a 3-carbon molecule. So 1 mole of glucose will give you 2 moles of pyruvate. And in the process, it produces this molecule called ATP, which is adenosine triphosphate. So this is the structure of ATP right here. These phosphate bonds are very high energy bonds. And so this is also considered as the energy molecule of the cell. So fermentation is actually an important pathway for the cells to produce energy from the sugars that they consume under anaerobic conditions. Pyruvate is then converted to ethanol and it releases carbon dioxide as a byproduct. This is a schematic of the entire process of bioethanol production in the industry. So in the previous slide, I showed you how you can take glucose, which is a simple sugar, and convert it to ethanol. But in the industry, they don't use simple sugars because it's very expensive to start from there. They typically use feedstocks like corn, sugarcane, or right now they're trying to move towards non-edible feedstocks, which is different types of agricultural waste. So you start with biomass and you need to pre-treat it. This pre-treatment is really important and it's probably one of the most challenging aspects of biofermentation. After pre-treatment, you get a mixture of different polymers, mainly cellulose, hemicellulose, and lignin. These polymers are then hydrolyzed by the action of different enzymes to break them down into their monomers, which are different sugars. So after the hydrolysis step, you get these simple sugars, C5, so 5-carbon sugars, 6-carbon sugars like glucose. which is then moved on into a fermentation reactor, where your microorganism will catalyze the reaction of glucose to ethanol. So a lot of the processes in the industry now use something called consolidated bioprocessing. This is where you try and combine all of these different unit operations, including hydrolysis of your polymers, fermentation, also pre-treatment in some cases, into one single unit operation. So this is often done to lower the processing cost, although it's really challenging. So here in this schematic, we've tried to combine the hydrolysis of the polymer and the fermentation in one unit operation reactor. And the microorganisms that are responsible for producing ethanol are engineered using genetic engineering strategies as well as molecular biology techniques. So this is the part that we focus on in our lab, and this is what I'm going to be talking about for the rest of my presentation. But this is just to give you an overview of so what we do is just a really small part of a big picture. There's a lot of upstream processes and downstream purification that need to also be done to make it industrially economic. So the second example that I want to show is insulin. So it's an anti-diabetic drug, and this is the structure of insulin. So it's a protein, complex protein, and in 1920s, the insulin that was used for treatment was animal sourced from pigs. And as you can expect, there was a lot of immune response because it came from animals. But until 1974, when recombinant DNA technology was first discovered and developed, there was no means of bulk production of insulin. So in 1982, Genentech, they developed the first biosynthetic process for human insulin production in E. coli. So this was actually the first human protein to be produced using recombinant DNA technology. And today it's predominantly produced either in E. coli or a yeast called Saccharomyces cerevisiae. The picture here at the bottom is actually a precursor of insulin. So insulin has two chains. This is an amino acid peptide. So it has two chains, chain A, which has 21 amino acids, and the chain B that has 30 amino acids. And you can see that they're linked by these disulfide bridges, which keep them together. The precursor of insulin has a linker peptide, this yellow C peptide. And when you cleave it, you have the active form of the drug. So again, this is a schematic of how the microorganism of the bacteria has been engineered to produce insulin. So for ethanol, I showed you a bigger overview of all of the processes involved. But for insulin, I'm showing you the specific processes that we use in our lab to make this ideal microorganism to make our compound. So for insulin, I'm going to cover each of this in more detail in the coming slides. So just bear with me a little bit. It might be a little too technical. So you start with a human pancreatic cell that has genomic DNA. So one part of this DNA is going to be the gene that produces insulin. So you want to isolate this specific gene that makes insulin, and then you want to put it into a plasmid. So plasmid is a circular DNA which is used to deliver enzymes into the bacteria. So this is a circular recombinant DNA, which then is inserted into E. coli or your bacteria. After the cells take up your plasmid, it now has the human insulin-producing gene in it, and every time your cell multiplies, it makes copies of your plasmid. So now you have in your fermentation tank, you have millions and millions of cells, each of them with a capability to produce insulin because of the plasmid that they carry. And finally, you do extraction and purification of the insulin to get your final product. So this is a more simplified version. It's a block flow diagram of the different processes that we use in our lab to go from concept to actually getting the product in hand. So after we know our substrate, which is our reactant or the starting material, and we know what is our target compound that we want to make, we look at the structures of these two compounds and we design the pathway. So this is includes identifying the series of reactions that need to happen to take it from the sugar to the chemical. And after you identify the reactions, you also need to identify the enzymes that would carry out each and every reaction. So this step here involves a lot of literature review and a lot of database search. So we go into databases to look at what reactions are possible and what enzymes can catalyze those reactions. Once we know what enzyme we want, we need to make those enzymes. So this is the second step called gene amplification. So here we make multiple copies of the DNA or the gene that has the information to make the enzyme. So we don't actually make the enzyme here, we make the DNA that would then make the enzyme when you put it into the cell. So this is called gene amplification. Moving on, the third step is plasmid cloning. So as I mentioned in the previous slide, a plasmid is a circular DNA. So you can think of it as a vehicle that you use to deliver the enzymes of your pathway to the cell. So this plasmid will have all the enzymes that you need to take it from the sugar to your chemical. And then you need to actually put the plasmid into the cell, and this step is called transformation. So at the end of these 4 steps, you now have a microorganism or a cell that has all the enzymes needed to take the sugar to the chemical. So now you do your fermentation reaction where you feed it with the starting material, you provide it with nutrients to grow, and you wait for about 48 hours for the reaction to carry out. After fermentation, we typically use HPLC or LCMS to do analysis of the media. So for most compounds that we produce in our lab, the cell would secrete the compound into the media. So after your fermentation, all of your media actually has your target chemical. So you centrifuge and separate the cells, and the supernatant, which is your media, will have your compound of interest, and you analyze it using different chromatographic techniques. So looking at the titer or the concentration that you get of your target compound, you might need to do optimization to make it better. So you do several iterations of this process until you have a high enough concentration that you can purify and use for downstream reactions. So optimization involves protein engineering, where you modify your catalyst or your enzyme to either make it more active or to make it better, make it a better binder to your reactant. You can also do metabolic engineering, where you eliminate all of the competing reactions in the cell. So you increase your flux of carbon from your starting material to your target chemical. So I'm going to go over each of this in detail over the next few slides. But before that, I just want to overlay this block diagram on the insulin schematic that I showed in the previous slide, just so you can get a better idea of how it works. So this is the insulin schematic. So this process here is gene amplification, where you isolate the human insulin gene from the genomic DNA. This is the next step called plasmid cloning, where you put it into a plasmid. And then this is transformation, where you put the plasmid into the cell. Finally, you do fermentation, where your actual reaction occurs. And then the last step in most industrial fermentations is purification of your final compound. So this is just to show you how these different processes play into the final picture. So the first step is pathway design. Here, you want to design all of your reactions that can take your sugar to the final chemical. Just as an analogy, if you look at organic chemistry reactions, an alkene can be converted to several different compounds using different reactions and different chemistries. And so you can decide which reaction you want to carry out depending on what is your intended product. And also, if you want to convert an alkene to an alcohol, you again have an option of using different catalysts and different reaction conditions. So this is very similar to what we do in our lab. So you have this metabolic network of tens of thousands of reactions, and you can exploit any of these reactions to take your starting sugar, say glucose, for example, to your final compound. Again, similar to chemistry reactions, you have an option of using different enzymes for doing the same reaction. So if there's a decarboxylation reaction, you have an option of hundreds of decarboxylases, and you need to figure out which is the best enzyme for your pathway. So just to give you a quick example of how we shortlist the enzyme we want to use. So say you want to take your starting material glucose to this particular compound here, and you identify that it needs 8 steps to go from the sugar to the target compound. And suppose your 7th step here is a dehydratase reaction. So this water molecule is dehydrated to give you this alkene bond. And the enzyme that catalyzes it is called an enoyl-CoA dehydratase. So if you look at the database, you will have at least 10 different dehydratases that you can find from different microorganisms. So how do you shortlist, or how do you pick your final enzyme? So some of the ways we use is looking at the kinetic parameters. So how active your enzyme is, how well does it bind to your substrate, and also to look at its natural substrate. So if I have an enzyme that naturally acts on this particular product here, I would definitely prefer using that enzyme over a different dehydratase that works on something similar but not exactly this compound. So those are some of the things that we think about when we finalize the enzymes we want to use. The next step is gene amplification. So this is where we make copies of the DNA that make the actual enzyme. So this is using a technique called PCR, or polymerase chain reaction. So you start with your genomic DNA. So this is a genome. It has all the enzymes or all the genes of the cell. And you can see the blue highlighted section, that's your target sequence. So this section is what has all the information to make your particular enzyme. So you want to make multiple copies of this target sequence so that you can then make enough enzyme in the cell. So the way this PCR works is that in the first step, it's called denaturation. So DNA is a double-stranded molecule. So in denaturation, you separate the 2 strands into single strands, and this occurs at a high temperature of about 95 to 98 degrees Celsius. After they're separated into single strands, there are things called primers. So the light blue lines here, they're called primers. So these are synthetic DNA that we design, which is complementary to this specific target sequence. So what I mean by that is, if I add these primers to the genomic DNA, it will search the entire tens of thousands of base pairs, and it will only go and bind to your target sequence because it's complementary to your target sequence. So this is called annealing, where your primers that you have designed will bind to your target sequence. And then the final step is extension, where your enzyme called DNA polymerase, it elongates this primer and makes multiple copies of your target sequence. So these 3 steps make up one cycle of the PCR reaction, and we typically do about 30 cycles in our reactions. And after one cycle, you get 2 copies of the target sequence from one. And if you keep going, it's a geometric progression. So after n cycles, you get 2 raised to n copies of your target DNA. This is the machine that we use for doing our PCR reactions. It can control temperatures, it can do temperature gradients, it can also control the time. And typically we take about 2 to 3 hours to do one PCR reaction. These reactions are really small volumes, so it's only about 25 to 50 microliters. And some of the important components that we add in this mixture is the genomic DNA. Again, this is the genomic DNA that has your target sequence that you want to make copies of. The primers, which is what you design to specifically bind to your target sequence. The polymerase enzyme and the nucleotides help to elongate the primer. And then finally, the buffer is used to maintain the salt concentration and the pH and other conditions in the PCR reaction. So now you have your, at the end of the PCR reaction, you have this really tiny tube with 50 microliters of a liquid. But how do you know that that's your target sequence? It could be anything else. So how do you verify that it's your target sequence? So there are two ways to do it. The first one is called gel electrophoresis, where you can only verify the size of your sequence. So if you know from the literature that your DNA should be 1,000 base pairs, using this technique, you can make sure that it's 1,000 base pairs. So DNA is a negatively charged molecule. So when you do gel electrophoresis and apply electric field, when you load your DNA on the negative electrode, it would start migrating towards the positive electrode. And this migration speed depends on the size. So a larger size DNA would migrate slower. And so at the end of it, you can look at the pattern under UV light. And if you compare it with a marker, so like a molecular weight ladder that has different sizes on it, you can compare it to the ladder and determine the size of your target sequence. And so this is one way that you can make sure that your size is correct. But of course, you also need to make sure your sequence, your actual target sequence is correct. And we don't do this in the lab. We outsource it and we send it for sequencing to an outside company, where they do something similar to PCR, and in about 2 to 3 days, they are able to give us the exact sequence of our target DNA. So they can tell us exactly if it's ATC or ATG and how it goes for about 1,000 base pairs. So you can take the sequence that you get, the results that you get from the sequencing, and you compare it with the literature sequence and make sure that they match perfectly. So the next step, so we finished pathway design. I then talked about making copies of the gene. And the next step is plasmid cloning. So this is where you put the gene or your DNA into the plasmid. So plasmid is a circular DNA. The three main components of a plasmid, the first one is origin of replication. So this is to make multiple copies of the plasmid. So every time your cell divides, you also want to make sure your plasmid is making copies of itself. So if your cell has a plasmid and it divides, the daughter cells will also have the same plasmid. So this this fragment here makes sure of that. The second component is the selection marker, and it's a really important component of the plasmid. So this is a way to ensure that your cell has the plasmid in it. So it's typically an antibiotic resistance gene. So say your plasmid has a resistance to a specific antibiotic, and if you grow your cells in that antibiotic, only the cells that have the plasmid and hence the resistance would be able to grow. So this is one way to make sure that the cells that you use have the plasmid and consequently have the enzymes that you need. And finally, you have the plasmid would have the different enzymes for your reaction. So in this schematic here, I've shown 2 enzymes, but it can hold up to maybe 4 or 5 enzymes depending on how big each of the enzymes are. So once the cells have these plasmid, they should have all of the enzymes that can carry out each of the reactions that you designed. So the next step is to put the plasmid into the cell. So this is called transformation, and it relies on permeabilizing the cell membrane. So there are 2 ways to do it. One is electroporation, where you shock the cells with an electric field. This creates pores on the membrane of the cell, and the plasmid then can enter through these pores. The second method is calcium chloride treatment, and it works in a very similar way, but it relies on heat shock. And you can see that the transformation efficiency is actually really low. Only about 1 in 2,000 molecules of your plasmid actually get into the cell. So this is why you need really high concentration of your plasmid so that you end up at least having a few cells with a plasmid in it. So this shows you how the transformation works. After electroporation, you have the plasmid inside your cell. And now every time your cell divides, it has a plasmid in it. And finally, you select the cells on an antibiotic plate. So you want to make sure your plasmid has a resistance. And so the white dots that you see here on the plates are all of them are different cells or different colonies, and each of this has a resistance which makes it grow on this antibiotic plate. The next step is fermentation. So again, if you look at the plate that you have with different colonies on it, you can be sure that all of these cells have your plasmid because they're able to grow on the antibiotic, and hence all of them have the enzymes needed to convert your sugar to your target compound. You then grow it in a culture, liquid culture, overnight, and then you do a fermentation reaction. So typically we start with a 5 ml fermentation in a 125 ml flask, and we carry out the reaction for about 48 hours. We control the temperature at about 30 degrees, and we constantly have agitation so that the cells don't settle down. And finally, after 48 hours, we do analysis using HPLC. So one of the most important things in fermentation is to design the media or formulate the media. Some of the important components of the media is a carbon source, which most of the time is glucose in our case, a nitrogen source, and we use yeast extract, which is a complex mixture of different amino acids. We also have a lot of salts in our media, and this is important because these salts provide important metal cations that are needed for the growth of the microorganism and also for a lot of enzymes that depend on these metals for their catalysis. And finally, we also have antibiotics. Again, these antibiotics would correspond to what resistance you have in your plasmid. After the fermentation reaction, as I said, you separate the cells from the supernatant, and for most cases, at least for all of the cases that all of the compounds that we produce in our lab, our compound is going to be in the supernatant. So it's not inside the cell, but it's in the media. So we analyze the supernatant using HPLC or LCMS, and we calculate their concentrations or their titers using area under the peak. The last part is process optimization. So as I mentioned before, a lot of the times after you go through all of these steps, you might just end up getting a few milligrams per liter of your target compound, which is really low for any practical purpose. So you need to improve the process using different techniques. And so these are some of the techniques that we use in our lab to improve the process. I want to mention here that you don't really need to go through the entire process and see that you have a low concentration to use these techniques. You can preemptively use this before you even do your fermentation. So one of the techniques is metabolic engineering, where you eliminate competing reactions. So say you have this pathway where this compound A goes to compound B. In E. coli, all of these intermediates can go be consumed by other competing reactions. So you want to remove all of those competing pathways by doing gene knockout. So gene knockout is where you remove the gene that codes for the enzyme that catalyzes your competing reaction. Yeah, and then this is a way to reduce byproduct accumulation in the cells. The second method is metabolic flux analysis. We don't do a lot of this in our lab. It's a computational method where you look at stoichiometric models of metabolism, and they use isotopically labeled substrates. So using these isotopically labeled substrates, you can find out the flux to different pathways. So you can see how much of your compound is going through pathway 1 versus pathway 2 versus pathway 3. And finally, we also use protein engineering, where we want to improve the activity of our catalyst or our enzyme, or improve the binding of our catalyst to the substrate. And then the last step in the industry is purification. We don't do a lot of purification in our lab unless for a lot of CFP projects, we do purification so that you can polymerize it. But in the industry, fractional distillation is used, is a very common technique. It's used for bioethanol purification. We also use chromatography, different types of chromatography like affinity chromatography, ion exchange chromatography. And finally, solvent extraction is also used in certain cases. So that was the end of section 1. Yeah, it was a little long, but I just thought it all fit together in one section. So I'll be happy to answer any questions that you may have at this time. Okay.
Please go ahead.
So section 2, I want to show how all of these processes that I explained in the previous section, how I have applied it to a particular example of biosynthesis in the lab, 1,4-butane-diol. So I'm hoping that this would make everything a little more relevant to what we do. So, okay. So for this project, we wanted to make 1,4-butane-diol. So it's a 4-carbon diol, and we wanted to start from xylose, which is a 5-carbon sugar. So the first possible route that we found in literature was this pathway that was used in 2011. So this here, your pentose or your xylose would go through the glycolysis, it would go through TCA cycle, and it would make this intermediate called 2-Kg or 2-ketoglutarate, which then after 4 steps or 5 steps, I should say, would make 1,4-butane-diol. So this entire pathway from your sugar to your final compound had 18 reaction steps. So this is possible, people do this, but we wanted to look at shorter pathways using lesser enzymes and having lesser burden on the cells. So we kept looking, and we came across this a very unique alternative pathway to make 2-Kg, the same TCA intermediate that I showed previously, from xylose. But now instead of 14 steps, we now have just 5 steps. So in these 5 steps, xylose can be converted to 2-Kg, and then you can use the same downstream 4 or 5 steps to convert 2-Kg to butane-diol. So this reduces the number of reaction steps significantly to just about 9 reaction steps. The one thing that I want to mention here is that these 5 enzymes here that take xylose to 2-Kg is not present in E. coli. So these are not enzymes found in E. coli. They are found in this organism called Caulobacter crescentus. So if we want to use E. coli to do this reaction, we have to take the gene from this organism. So use the genomic DNA of this organism, get the genes, make the enzymes, and put it into E. coli. We also wanted to look at further reducing the steps or making it more manageable. And so when we looked at the structures, so if you look at the structure of this compound, it's the intermediate before 2-Kg in the pathway. You can see that in just 2 steps, so a decarboxylation step to remove the CO2 here, and then a reduction step to convert both the aldehydes to alcohol, you can get butane-diol. And so now you can convert your sugar to your compound in just 6 reaction steps. So here, again, the first 4 enzymes is from this organism called Caulobacter. Since decarboxylation and dehydrogenation are very common reactions, and you have different organisms and you have wide choice, so we tried decarboxylase from 3 different organisms and the dehydrogenase from 5 different organisms, and then we did experiments and then finally chose the enzymes that gave us the highest concentration of butane-diol. So in this project, we did metabolic engineering before moving on to fermentation because we knew that E. coli has certain enzymes that would take away the carbon from our pathway. So xylose itself in E. coli can be isomerized by this enzyme. So we removed this gene from the genome of E. coli. We also removed these 2 genes, YJHH and YAGE, which can metabolize xylonate. So our final strain that we have is this strain here, where BW25113, that's the designation for the wild type E. coli strain that has all the genes intact. And then we did 3 gene knockouts, so we removed these 3 genes. And so this is the final strain that we used for our experiments. So once we do metabolic engineering, we move on to plasmid cloning. So in order to do the plasmid cloning, you need the actual DNA fragments of each of these 6 enzymes. So we did PCR, which is gene amplification using the genomic DNA, and we got the DNA for each of these 6 different enzymes, and then we put them into 2 different plasmids. So the first plasmid had 4 enzymes, all the 4 upstream enzymes from Caulobacter, and the second plasmid had the decarboxylase and dehydrogenase. So again, since we tried different combinations of decarboxylase and dehydrogenase, we had multiple of these plasmids, depending on what we wanted to choose. Once we have both the plasmids, we then transformed it into this particular strain that has these 3 gene knockouts using electroporation. And finally, we selected on ampicillin and kanamycin. So both of these are 2 different antibiotics. And the reason for choosing these 2 antibiotics is if you see the plasmids, the first plasmid has a kanamycin resistance, and the second plasmid has an ampicillin resistance. So if your cell takes up successfully both of these plasmids, it should have both the resistance, and hence it should be able to grow simultaneously on a medium that has both these antibiotics. So you can be sure that the white dots or the cells that you have on this plate, they have both of your plasmids, and thus they have all of your 6 enzymes needed to convert xylose to butanediol. The next step is fermentation and metabolite analysis. So this is a picture that I used in the previous section. So you pick the colonies from a plate that has both the antibiotics, you grow it in a liquid culture overnight, and then do a shake flask fermentation followed by analysis using HPLC. This trace here is a typical HPLC trace that you would see from our fermentation media. So I'll just go over some of the results of the project. So this is where we screen different enzymes. So the first one we screened was different decarboxylase, which can remove this carbon dioxide here and make butane dial. We tried 3 different decarboxylases from 3 different organisms, and we chose the one that gave us the highest concentration of BDO using the HPLC integration under the peak. Similarly, we tried 5 different dehydrogenases, and again, similar to the previous screening experiment, we chose the enzyme that gave us the highest concentration of our final compound. So at the end of these 2 experiments, we now have the final 6 enzymes that would convert xylose to butanediol. But when we looked at the HPLC trace, we saw that this peak here around 21 minutes corresponds to BDO, our target compound, and it was about 1.8 grams per liter. But you can see that there's this huge peak around 14 minutes, which was about 3.6 grams per liter, corresponding to BTO, which is butane triol. And this was a major byproduct in all of our fermentations that we had done up until now. So the next objective that we had was to reduce the byproduct formation and improve the production of our final compound. So if you look at the pathway that we designed to make butanediol, we want to look at how that pathway resulted in byproducts. So if you look at the structure of this compound 2,5-dioxopentanoate and the intermediate before that 2-keto-3-deoxy-D-xylenate, they actually have a very similar structure on the left part of it. And so this decarboxylase enzyme KIVD, it's an enzyme that has a very broad substrate range. So it can decarboxylate a lot of different substrates as long as they have a similar structures. So we wanted to decarboxylate 2,5-dioxopentanoate, but if it decarboxylates the intermediate before that, you can see that the extra hydroxyl group here would result in our byproduct butane triol. So we now had to selectively reduce the activity of the enzyme KIVD towards this undesired substrate and maintain or even better improve its activity towards the desired substrate. So this is where protein engineering comes into picture, where you look at the crystal structure of the enzyme and you manipulate the crystal structure so that it improves its activity. So this is the crystal structure or the binding pocket of KIVD, where your actual substrate would bind to the enzyme. And these are the 2 competing substrates. So the one on top is the undesired substrate, and you can see that the extra hydroxyl group makes it bulkier, so it's bigger, and the one on the bottom is our desired substrate. So the idea behind this engineering was that in order for your substrate to bind with the enzyme, it needs to enter the binding pocket. So we thought that if we shrink the binding pocket, make it smaller so that the bulkier undesired substrate won't be able to enter the pocket, we can reduce the undesired byproduct activity. So we shrunk the binding pocket by making the amino acids lining the pocket bigger. So as an example, if we had a serine in the original enzyme, which has a hydroxyl group, we replaced it with phenylalanine, which has an aromatic ring. So since OH is now replaced with an aromatic ring, it occupies much more space, and thus it makes less room for the substrate to bind. So using this idea, we constructed 10 different mutants. So we did 10 mutations. And one of these mutations gave us really good results. So we were able to reduce the byproduct to less than a gram per liter, and we improved the production of butanediol to about 3.8 grams per liter. And this was again after integrating the peak under the HPLC. So the last step is to scale up. So typically, if we get decent titers in our shake flask fermentation, we then scale it up to a 1 liter bioreactor to see how much we are able to obtain. And for this process, we were able to get about 10 grams per liter BDO in 36 hours from xylose. So the way we operate a bioreactor is different from shake flask fermentation. So in the shake flask, which is like the really small scale, everything is done at the same temperature and same conditions. But in a bioreactor, we have two different phases. We have a growth phase where we first let the bacteria grow. So the temperature is higher, it's about 37 degrees, which is optimum for cell growth. And after the cells reach a particular density, so after we have enough amount of cells, we then lower the temperature and allow it to produce our specific compound. So we lower it to 30 degrees, which is ideal for production. And we also lower the dissolved oxygen level to 10%. And the bioreactors that we use, they are equipped so that they can control the temperature, the dissolved oxygen, the pH, and also the agitation. And we typically use it in a fed batch mode, which means that the glucose that we feed for the cells to grow, we only add it depending on how fast the cells consume it. And this is really important because you don't want the cells to have excess glucose, and at the same time, you don't want to starve them. So you want to maintain it at a particular concentration. So we sample the fermenter every few hours, and we do the feeding dependent on how fast the cells consume the glucose. So that was the end of section 2. I'll be happy to take any questions. Okay, so my last section is, it's just a couple of slides, and it's more to give you an idea of how industrial fermentation is different or similar to what we do in the lab. So the efforts made in the industry to improve concentrations or titers. And I'm going to use penicillin as an example because it's a really classical fermentation example on how fermentation has improved over the years. So this is a structure of penicillin. You can see that it's a cyclic compound, and it's a little complicated. So I'm sure everyone is aware of or have heard of the Fleming's discovery in 1928, but not a lot of us know the amount of efforts and the time it took to go from being discovered to being bulk produced in the US. So this is actually the first antibiotic to be produced in bulk, and initially very small quantities were being produced, and it was very unstable at room temperature. And the microorganism that produces it is a Penicillium species. It's an aerobic fungi that needs oxygen to grow. The other thing to note is penicillin is a secondary metabolite. So what that means is the synthesis of this compound is not linked directly to its growth. So when your microorganism is growing, it's not producing your compound. And then when it stops growing, it will start producing your compound. So it's not linked to each other. So today, the worldwide sales is about $15 billion a year, and the cost has dropped from $300 per kg in 1953 to $10 per kg. And you can see this graph here that shows the increase in the titer from like 1 milligram per liter to today it's about 50 or more grams per liter. So early penicillin production, they used something called surface fermentation. And this is because, as I mentioned, the strain that they used is an aerobic strain that needs oxygen to grow. So they wanted to maximize the surface area and not have a lot of depth so that all the cells and every point of the fermentation tank, they have enough oxygen to grow. So they use surface fermentation about 2 centimeters deep. The carbon source that they used was sucrose or glucose, and sodium nitrate was used as a nitrogen source. And the yield that they got, or the concentration, was about 1 milligram per liter. So some of the problems with this initial production was, one was that the cells would grow rapidly with absolutely no production, and then after all of this sugar was consumed, they would start producing penicillin, and the amount of time that penicillin was produced was really minimum, so it was only over a span of 2 days. And also you can see that if you look at the pH graph, it dramatically changes over time, and the optimum pH, which is around 6.5 to 6.8, was only maintained over, again, a span of 2 days. So it was very difficult to stabilize the pH and also have enough time for production of penicillin. So the first change that was made was to change the carbon and the nitrogen source. So the carbon source initially was sucrose or glucose, which is a very simple sugar. It can be very easily metabolized by the cell, so it releases carbon much more quickly. So they wanted to change it to something like lactose, which is more slowly used by the cell. So it's something that's more difficult for the cells to use, so it releases sugar gradually over time. This improved the time span of penicillin production from 2 days to over 5 days. And also they changed sodium nitrate and replaced it with ammonium acetate or lactate to stabilize the pH during fermentation. So they were able to handle the pH fluctuation much better. And this improved the penicillin production 5 to 10 folds. The second improvement was to use corn steep liquor. So this was actually a very fortuitous discovery. They were just looking for a better way to use corn steep liquor because it was initially just used as animal feed. It wasn't being used well. And so they tried using this as an additive for penicillin production, and they discovered that it was able to buffer the pH and facilitate slower release of sugars, both of which was essential for better penicillin production. And the more important thing was corn steep liquor actually provides the precursor penicillin G, which leads to a more stable form of penicillin. So using this, they improved the production further to about 50 to 100 milligrams per liter. Probably the most important change, I would say, was to change from surface fermentation to submerged or deep tank fermentation. So since it was an aerobic fungi, if you change it to a deep tank or submerged fermentation, you need to make sure that every point in the reactor, the cells have enough oxygen to grow. And so they needed to use constant agitation and aeration. And a lot of people say that this change to deep tank fermentation was almost as important as the discovery of penicillin to allow bulk production today. The last effort made was classical strain development. This is something that's very commonly used for a lot of processes in the industry, where once you identify a species, you try different strains of the same species to identify a better producing strain. So something that has intrinsically higher activity, better production, better growth. And they also subjected a different strain, Penicillium chrysogenum, to X-rays and UV light to produce mutants that were high-producing mutants. So using all of these different techniques, they were able to get the yield to about 50 grams per liter. So starting with 1 milligram per liter, they were able to go right up to 50 grams per liter. And some of the other modifications that are still being pursued today is to increase the level of transporters, so to help penicillin excretion out of the medium, nutrient absorption into the cell, and also to amplify the gene clusters that make penicillin. And then for the last slide, this is just a schematic of the bioreactor used for penicillin production. Depending on the depth of this bioreactor, you can have multiple impellers. There's temperature control using cooling water, a pH probe where pH is controlled with acid or base addition, and also a temperature probe here. Typically, the growth phase lasts for about 40 hours, after which it's switched into the production phase, which goes on for 120 to 180 hours for penicillin. And on the right, it's a schematic of how the process works. So the important thing I want to point out here is that 10 times in every 6 days, they would remove 30% of the culture to isolate penicillin and add 30% of fresh medium. So you want to maintain the same volume, but you want to sustained production. So this is a technique that's used in a lot of industrial fermentations where you maintain the same volume, but you remove the culture and supplement it with fresh nutrients so you can keep the cells growing for a longer period of time. And finally, they were able to get about 99.5% pure penicillin. And that's the end of my presentation. I would be happy to answer any questions that you may have.